Alrighty guys, we are going to finish up our notes on chapter 3 today, and you have the assignment in the box or in Canvas about turning in your chapter 3 notes. So everything that is written in blue in my notes should be written down in your notes as well. The black part I told you could leave out. And that's going to be part of your notebook check, which will go towards a very big grade at the end of the 9 weeks. Usually they were somewhere around, 80 points. So it might only say 6 points on there, but the vocab adds up and the amount of notes that we do adds up as well. So these are really easy points to get. Hopefully you are doing them and not just brushing it off to the side. So right now we're going to talk about proteins, and proteins can be nuts, eggs, dairy things, meats. Here's some fish, some beans. Proteins are made up of carbon, hydrogen, and oxygen, and nitrogen and occasionally sulfur. The monomer of a protein is called an amino acid. I'm not having you write that down because you guys know that the monomer of a protein is an amino acid based off of your vocab terms. These are all the different proteins. Well, these are some of the different proteins. Some of them are nonpolar, meaning that they cannot be dissolved in water. Others are polar, meaning that they can be dissolved in water. And the part that you see highlighted in blue, or what is like a grayish color, those are going to be the variable groups. So amino acids are going to consist of a central carbon atom, a hydrogen atom, and an R group, along with a carboxyl group and an amino acid group. Now the R group, or the variable group, could be any of 20 different functional groups. That is a lot of functional groups, but on the same note, it's not that many functional groups because you guys all look very different, but you all have the same exact 20 different functional groups to work with. They're just put together in different orders. I'm not going to have you draw the amino acid structure because, well, it's hard to draw online, but you can just observe it here. This is the central carbon group right here. Here is the carboxyl group. Here's the hydrogen. Here's the amino group with the N, which is nitrogen, and the variable group is right here where that R is. To give you an idea of what this would look like in an actual amino acid a little closer up, we have our central carbon with our hydrogen on both, the amino group, the carboxyl group, and here would be that R group, but instead of the letter R, we have a hydrogen in there. That makes glycine. If we replace that with a CH3, a carbon and 3 hydrogens, now we have alanine. So simply by changing just one little element, you end up with completely different structures in amino acids. A dipeptide is formed from the condensation reaction of 2 amino acids. Condensation reactions are going to be adding things together. So the prefix di means 2. That's how this makes sense here. 2 amino acids give us a dipeptide. A polypeptide is formed when many amino acids are joined through a series of condensation reactions. So you know condensation is putting things together. The prefix poly means many, so you're putting many things together. Proteins are going to be composed of 1 or more polypeptides. So polypeptides are really big, and we have to have a bunch of these in order to actually get a protein. Proteins often assume a 3-dimensional structure as a result of interactions such as hydrogen bonding. So these are the different shapes of proteins, and you don't have to write any of this down. The primary shape is going to be a chain, which is just a linear sequence of amino acids. You could have a secondary shape, also known as a beta sheet, which is going to be a folded sheet, or you could have an alpha helix, which is a spiral shape. Tertiary is the lowest energy state with the maximum stability, and quaternary is a glob. It's multiple proteins interacting with one another. So here is the difference between like a primary structure, which is very simply linked, and a glob that's just a big mess. If you ever go to the gym, or if you know someone who goes to the gym, they might take a protein supplement. And protein supplements, they vary in the amount of protein they contain, but you want to make sure that you're only taking a serving. If you go in there, you work out really hard, and you come home and you take half of a tub of protein, your body is not going to be able to use it. So just because you're taking it doesn't mean that it's actually doing anything for you at all. In reality, your body is just going to use what it can, and then it's going to get rid of the rest. How do you think your body gets rid of stuff that it doesn't want anymore? Yeah, you're going to get rid of it that way. Alrighty, moving on. A protein can be denatured. Oh, that word sounds awfully familiar. I think we talked about this in chapter 2 when we were discussing catalysts and enzymes. So if a protein is denatured, it's going to result from a change in its three-dimensional shape. This could be caused by change in temperatures, the type of solvent it may be dissolved in, or by pH. We already talked about changes in temperatures and pH with changing enzymes and catalysts. So, do you think a protein is an enzyme? Here's what it should look like. And you can see this is an active site in here. After it got denatured, maybe a temp change, or maybe it was too acidic. Oh, there's no active site anymore. It's just a little mess. An enzyme is a biological catalyst and is essential for the functioning of a cell. Oh my goodness, enzymes are back. Like a catalyst, an enzyme may be reused. The effectiveness of an enzyme depends upon the physical fit between it and the substrate. So here we go, just to review with you, you got your enzyme down here. Here's the active site and the substrate. When it fits into that active site, the chemical reaction is going to be taking place. We have an enzyme substrate complex. And as the chemical reaction is finishing up, that substrate will then be popped off and released, and you will have your product. Sometimes it's going to be splitting things apart. Other times it'll be combining them together. Alrighty, last part here is talking about nucleic acids, which is the fourth and final macromolecule. Here is a nucleic acid of DNA. These are very large and complex organic compounds that store genetic information. They use a sequence of 4 nitrogen bases arranged in a certain order to store hereditary information. Again, don't worry about drawing it, just observe. So you have a sugar deoxyribose base. It's a 5-carbon sugar base with a phosphate group, and then there is a nitrogen base over there off to the side. DNA and RNA are both examples of nucleic acids. Now, the monomer of a nucleic acid is called a nucleotide. This is made up of a 5-carbon sugar, or pentose, a phosphate group, and a ring-shaped nitrogen base, which was all those shapes that I just showed you a second ago. Nucleic acids are, there's not much to them. It's pretty quick and to the point. I do want to review condensation hydrolysis reactions really quickly here. So condensation, remember that's joining 2 molecules by a covalent bond to make a much more complex molecule. This is also known as dehydration synthesis because you are releasing water. So you are dehydrating the object, essentially. Here you go with your condensation. You have 2 separate pieces, whether monosaccharides or amino acids, and we are going to be pulling off a hydrogen from one and an OH from the other one. That's going to give water. That water is going to go off to the side. We've dehydrated both of those, and now they are combined. Hydrolysis is breaking a bond in a polymer into monomers, and water is going to be used here. So here is my polymer. I'm going to try to split, I guess, right here between the C and the D. I want to break that bond, and in order to break that bond, I have to give that C and D something else to adhere to. So I'm going to add water in, and now that bond is broken. Alrighty, guys, that is it for our notes. We are now finished with Chapter 3. Remember, you have a vocab quiz next Tuesday on the Chapter 3 terms, and we're going to kind of work with this material a little bit before we get into talking about a Chapter 3 test. Remember to turn in your notes. Talk to you later.